in medical school and the PhD from University of Copenhagen, Denmark. So his research right now working on the DNA repair and the connection between nuclear genome and mitochondrial genome and their contribution to age-related disease, especially like neurodegenerative disease. So I think that Dr. Wilhelm Bohr got a lot of interesting work, and also without further ado, I'll just reduce my introduction and let him to tell you about the very exciting stories. Also.
All right. Well, thank you very much for having me in this introduction. And first of all, can you hear me well?
Yes.
Can people hear me?
We can hear you. Yeah.
And see the screen also? Can you hear and see?
Yes.
Because we did have a little bit of a problem before with the sound, didn't we? So it works well now? Should I just go ahead and start?
Yes, perfect. With sound
right,
and the image, yeah.
Yeah. So I first of all, I'm pleased to talk to this group, and it's exciting with that new mitochondrial journal, and I look forward to publishing there with time and to working with you guys a little on that. And then it's also a pleasure for me to talk together with Evandro Fang, one of my favorite postdocs, and we will give a shared seminar. I will give a more broad sort of background to many of the things, and Evandro will give a little more recent work that he's done in his new lab in Oslo, where I enjoy to visit him from time to time. So I'm going to talk about aging, DNA repair, and mitochondria, and a couple of things, and I'm going to give a broader view of things we've worked on for a number of years. And as you know, we are now interested in these so-called hallmarks of aging, which is a consensus formation in the underlying causes or topics of aging. And here you'll see that genomic instability is a major one, mitochondrial dysfunction a major one, and I think that in cellular senescence and other things, but that this has contributed to that now people in the field of aging and many related fields, cancer and other things, all agree that mitochondrial dysfunction, aging, and neurodegeneration and cancers. And so I've been interested in the aspect of DNA damage, DNA repair for quite many years. And here you see that when we are surrounded by things that we all are surrounded by, including air pollution that we talk about on problems on a daily basis with that, mitochondrial rust formation, dietary intake, all of these things cause damage to the DNA and also to other macromolecules. But the damage to the DNA is probably more lasting than the damage to the other macromolecules, who can more easily be degraded and reconstituted. So the DNA damage has become a very important thing in many areas of research. We have many kinds of damage to DNA, and these are just some specific lesions. Is there a problem? Somebody's talking. Is there a problem? Is it okay?
No, we can hear you, but 每个人请那个 mute. Please mute yourself.
Okay.
Everybody.
Please mute unless you want to say something. So anyhow, there are these more established types of DNA damage that many people have worked with for many years, but also we know that there can be many other alterations in DNA that are not exactly a base damage such as these are. And we have many thousands of these lesions per cell per day, and they accumulate over lifetime. So this is a very big problem in biology. And here I just much like that. What I showed you, but could also be a DNA modification, structural change in DNA that I'll talk a little bit about, but also it signals a lot of other processes, and the endpoints of these processes are senescence, aging, neurodegeneration, and cancer. So when you are damaging DNA, it causes a lot of changes, direct effects on processes and modifying chromatin structure and many things, a very broad signaling process that causes a lot of changes in the cell. And what we have worked with over now the last 10, 15 years, and where Evandro Fang has played an important role in finding this process, is this system here where we have a pyrrolation signal that leads to mitochondrial dysfunction. And this is something that I'll talk about. I think now it's gained traction that this could be one of the major aspects of what causes what the PARP1 is the most important part. Protein has many functions in the cell, and there's a lot of interest in it currently. I was just at a very big meeting where half of the talks were about PARP, and we know that PARP is, by the way, one of the first proteins that reaches DNA damage if it's a DNA double-stranded break, for example. But I hear background is this, if there's something wrong, please interrupt me or something so I know. But okay, so we have a PARP1 is involved in the DNA damage response, but also in the pyrrolation process by which we form these big poly-ADP ribose chains at sites of lesions in DNA and DNA modifications. And this pyrrolation, which is a very central thing in our interest, occurs all over the place in many parts of the genome. And we are still at a process of trying to understand how much occurs in different components of the genome, how much pyrrolation takes place, say, after damage in different organelles and different compartments of the genome. And one of our interests is exactly what happens in mitochondria. And mitochondrial pyrrolation is an area of great discussion and great dispute. But most of the cellular NAD that is the substrate for the pyrrolation is in the mitochondria. And we and others have shown that peroxide or DNA damage increases nuclear pyrrolation and decreases mitochondrial pyrrolation. And so this has a lot to do with shifting of NAD pools between nuclear and mitochondria. And I know there are many Chinese groups interested in this, and I've written some very good reviews from Chinese groups in this area. And so one question is there mitochondrial pyrrolation? Is PARP1 in mitochondria? Is another part there that actually gets pyrrolated inside the mitochondria, and which PARP is involved? So I had in my former version of this talk that before today, I was going to show a number of very recent unpublished data on this pyrrolation, but now that this talk is recorded, I was told not to show unpublished results. So I will just suffice it to say that here in a mitochondrial highly purified prep, you see PARP1 here in the wild type. When we knock down PARP1, we don't see anything. So we believe this is one of our experiments that support the notion that PARP1 is present in mitochondria, and we've done a lot of work with this and using ADPR chip and CHAP to show that PARP1 binds to mitochondrial DNA and actually pyrrolates in the process. And we think that PARP1 may play a role in pyrrolating TFAM, which we're currently investigating. And this is all unpublished and unfinished results, just to show you something that we're really excited about right now, and that there's a lot of dispute about in the field because there are many people who do not believe that PARP1 is present in mitochondria. So a little more background on this. We have, of course, a very distinct genome in mitochondria and the nucleus. Nucleus very compacted, very highly organized genome, whereas in mitochondria, it's just this little plasmid. But interestingly, most of the mitochondrial proteins are encoded by the nucleus, as we know. And then we're all interested in signaling between the mitochondrial nucleus, and there are many signaling pathways that take place in that regard. And in the middle of all these things, between them, we have a very complex metabolism And amongst these, NAD is a very key molecule that serves in many aspects of this metabolism, both in mitochondrial nucleus and in transactions between the mitochondria and the nucleus. And so NAD pools are being eaten up, as I mentioned, by pyrrolation. So if you have DNA damage forming these large molecules of PARP here, we deplete NAD pools, and therefore we deplete functions that depend on NAD, and there are many functions that depend on NAD, notably sirtuins, PGC1 alpha, and regulators of mitochondrial function. And so if you lower your NAD substantially, it causes mitochondrial dysfunction. So our inroad into, there's some noise, our inroad into this aspect was that for many years I've been interested in working with these human syndromes where the patients appear much older than they actually are. We call them premature aging disorders. Werner syndrome is quite famous. Here you see a patient 48 years old, looks much older than they actually are. And in all of these diseases, we have a phenotype that looks much older than the biological chronological age of the patient. So we have used these as model systems to investigate aging, and many other labs do that as well, because it's a way where you can study more specific mitochondrial and molecular functions underlying these diseases, and then extrapolate that to aging. So they are good model systems for aging, and also they all are defective in DNA repair, suggesting some important role of DNA repair and protecting against aspects of the aging phenotype. And what we stumbled or were interested in was that in some of these diseases, we have severe neurodegeneration, and we were interested in whether that then could be associated with mitochondrial dysfunction. And at the time that this connection had not been made. We know that in mitochondrial disease, we have very severe neurodegeneration in many cases. And so we could make a connection between these disease phenotypes and mitochondrial dysfunction at various levels using various approaches, cell lines, mouse models, nematodes, bioinformatics, databases, and published a long number of papers on this, establishing this mitochondrial phenotype in these conditions of DNA repair deficiency that are associated with severe neurodegeneration. So we found that in these diseases I mentioned, and they all in common had increased pyrrolation, which is a sign of this DNA repair deficiency. So DNA damage accumulates in the genome and gives you this increased pyrrolation. We call it hyperpyrrolation. And this, we found increased mitochondrial dysfunction, increased ROS formation, membrane potential changes, and decreased mitophagy, and lower levels of NAD, decreased sirtuin activities, probably as a consequence of that. And also many clinical features of these diseases are similar to what you see in mitochondrial diseases. And here is a picture of a damaged mitochondrion commonly found in these conditions. More lately, we've expanded this to Werner syndrome and ataxia, and also in many aspects of Alzheimer's disease, which I'll talk about a little later. So this is just an example of this pyrrolation, hyperpyrrolation. Here's a mouse cerebellum, CSP, Cockayne syndrome minus here, and you see this very increased pyrrolation. These are worms with CSP. You see older worms and CSP worms have increased pyrrolation. We see it in ATM cells as well, which we also have worked a lot with. But interestingly, we don't see it in XPC, which is a similar DNA repair deficient disease without neurodegeneration. And we see lower levels of NAD generally in these conditions using various approaches to look at NAD levels. So it led us to this model here that we are working with, and it suggests that we have a DNA repair deficiency, either because of these diseases, or as we know, there's deficiency in Alzheimer's disease. I'll return to that, or maybe because of aging, where we also know that there is a decline of DNA repair with aging. Studies have suggested about 1% decline of DNA repair with aging per year. This leads to DNA damage accumulation in the nucleus here of the cell, increased pyrrolation, lower levels of NAD and NAD-dependent molecules, mitochondrial dysfunction, and then mitophagy is the main finding in these mitochondria, and Evandro Fang was very instrumental in making the connection to the mitophagy here, but we also have seen other aspects of mitochondrial health and dysfunction in various of these diseases that I mentioned. And we think the mitophagy may be a key factor of giving neurodegeneration in these diseases. And so we can do something about this. We can supplement with NAD because NAD levels are lower. And by the way, there's a lot of literature suggesting that NAD declines with age in general in normal human beings, but while it's very established in mouse models, it's not really solidly established what happens in humans, and it may be very dependent on individuals, how much NAD declines. But when we then complement there with NAD supplementation, nicotinamide riboside or NMN is what we mainly use. We stimulate mitophagy, we improve mitochondrial health, but we also improve DNA repair. And we and others have shown that NAD supplementation can stimulate DNA repair. So we have the NAD supplementation plays in a very important components of our model here. And mitophagy is a very complex, important process. Somebody's drawing something here on my screen. Somebody's drawing something on my screen. Thank you very much. But anyhow, there are many processes in mitophagy, and we're working on different aspects of different components like ruthenium-3 and other proteins to really establish mechanistic pathways in the mitophagy relating to the conditions that we're interested in. The main thing that happens in mitophagy is that the lysosomes fuse with the mitochondria and engulf these mitochondria and get rid of the bad mitochondria. And it's an essential process. If you don't have mitophagy, you accumulate bad mitochondria. Somebody's drawing on my screen here. Can you remove that, please, whoever did that? Somebody put some red lines on my screen.
Yeah, I don't know how to remove this thing. Somebody like in the audience by accident, I guess, but how to remove this?
Okay, all right. I'm not, okay.
I don't know if the host can do that.
Yeah, I don't know. I'll see if I have, if it goes on. It goes on in all my slides. I'll try to stop my talk and get back to the slide. Maybe that'll help. That's not very, I don't want to have that sitting there. Okay. So what, maybe that, so. Still there. Huh? Okay. I think so.
Reopen this slide and try it again.
Via, maybe you can quit the PPT and then open it again.
Okay.
Yes.
Sorry.
Good idea. It's okay. Now, that was weird.
I'm pretty sure it was an accident.
Yeah, of course it was an accident. Yeah, I don't. I haven't seen it before because I thought that when you do that, you have to ask permission from the speaker to draw on it. So Anyhow, that's okay.
So some
A
speakers
little,
also have such
we get a
situation.
little adventure in my speech action.
Exactly. So, yeah.
So, okay, let me get back to where I was here. So,
Like, we couldn't see your slides. You want to share slides again?
okay, why not?
So share slides.
Yeah. But I think it did remove this. But so, whoever it is, please don't draw on my screen. Thank you very much. Okay. Maybe so I mentioned these diseases, and I just point out that these disease molecules play in broadly in DNA repair in many pathways. So this effect that we find here is pretty broad in both base excision repair, nucleotide excision repair, recombination, double-strand repair, and so forth. And so one of the specialities of my lab over many years has been to characterize DNA repair processes in mitochondria. And I thought this might interest your group a little bit because there are not many labs that do this because it's very difficult to characterize DNA repair in mitochondria and make sure you have very pure mitochondrial fractions, do some biochemistry and other things. So it's been a very difficult area to work with and a bit controversial as well. But what we know is that here's the nuclear compartment, here's the mitochondrial compartment. And so the removal of oxidative lesions in DNA pretty much is similarly occurring in mitochondrial nuclear DNA. So here we have a number of additional base excision repair proteins in the nucleus, but we have, and some also we have interestingly some specific proteins in the mitochondria that are involved in base excision repair and not found in the nucleus. And here again is the both compartments. And we discovered some years ago, and another lab did too, that DNA polymerase beta, a key protein involved in base excision repair, in fact, is present in mitochondria. I'll talk more about that. Then the process of single-strand break repair is pretty much also present in mitochondria. Mismatch repair is different in the nucleus with different proteins than it is in the mitochondria. And nucleotide excision repair, the kind of repair that removes bulky lesions in DNA, is largely not present in mitochondria. All of these proteins here involved in nucleotide excision repair have not been found in mitochondria. Double-strand break repair is also not really present in mitochondria, although people have reported end joining in mitochondria. It's not really been solidly determined that these proteins are present in mitochondria. Then we have other DNA metabolism proteins present in mitochondria that are present in both compartments. So this is double-strand break repair is not really present in mitochondria. It's just a minus here. So when we put that together with our work in these diseases, we believe that it is the nuclear damage that signals what I just talked about, the signal process, and not a defect within the mitochondria caused by the absence of whatever protein it is. We're talking about disease protein not being present in mitochondria because some of these proteins are not present in mitochondria and some are. So when we see the exact same mitochondrial dysfunction in CSB and XPA, xeroderma pigmentosum group A, and in ATM ataxia, then that means that it's not due to whether or not the protein plays a direct role in mitochondria. Now in CSB, Cockayne syndrome, the protein does play a direct role in mitochondria, so it adds to it. But I'm trying to argue that the signaling process by which we get the mitochondrial dysfunction comes from signaling from nuclear damage. When we then supplement with NAD, we have a lot of very beneficial effects that we measure in these systems. We decrease pyrrolation and ROS, increase mitophagy, get higher NAD levels, sirtuin activities, and improve DNA repair. So this has led to, for us, to some clinical interventions that are ongoing. And it's been satisfying to see that we can do this after many years of doing cellular and biochemical work. So we truly have taken Cockayne syndrome and Werner syndrome from very biochemical cellular stages to clinical intervention. And we have a study in Cockayne syndrome and a study in Werner syndrome ongoing with NAD supplementation. So I'll talk a little bit about this disease, Cockayne syndrome, and make it pretty quick because we lost some time here. But it is a disease that's interested me for a very long time because I started working with something called transcription coupled DNA repair and many years ago, and this disease was deficient in transcription coupled repair. And now we think that the mitochondrial dysfunction is a major factor in this, but the disease has a lot of features and very severe disease for the kids that get this. And they don't get cancers, interestingly, but they get severe neurodegeneration. These are some clinical studies, and one of the studies, and many of the studies find these different features in the disease where hearing loss is a major one in both diseases, those studies. So the patients suffer from many features, visual features, but also a very significant hearing loss. And I think I'll skip a little over this, but CS proteins play major roles in many processes. The protein itself has a DNA-dependent ATPase activity and is involved in transcription and transcription-coupled nucleotide excision repair, has a helicase domain but no helicase activity. And so the major CSA and CSP proteins are two different proteins from two different genotypes of this disease, but they both play roles in DNA repair, RNA synthesis, transcription, and in this mitochondrial abnormality. So we find mitochondrial dysfunction both in CSA and CSP complementation groups in cells and mice, and also in chromatin remodeling. So our recent work suggested that from many viewpoints, we're looking at patients, looking at nematodes, the worms, mice, complemented cells, they all share a severe mitochondrial dysfunction, which can be improved at the cellular level by NAD supplementation. And again, I think I will just run a little over this to show you that. So we did some NAD supplementation in some mice a while back and found that mitochondrial parameters were greatly improved after NR with nicotinamide riboside NAD supplementation. So we saw that from we could improve the membrane potential to normal levels, the ROS to normal levels. So treating mice within NAD supplementation improved the mitochondrial function in these mice dramatically. And more lately, we've expanded a lot into hearing as a model system to study function and neurodegeneration, and then smelling, which I think is a really interesting feature that is very understudied. And we're trying to make connections now between smelling and specific neurodegeneration, mitochondrial dysfunction, and NAD supplementation. And in all of these cases, we see some effect of NAD supplementation, but I won't be talking about smelling at all. I'll talk a little about hearing. So in the human, we have the cochlea here, the inner ear, and that's where the waves go through here from the lower to the higher frequency. And in we used then the CSA and CSB mouse models, which had already been shown to have a hearing deficit, and looked at NAD levels. And in both CSA and CSB mice, we saw lower levels of NAD in the actual cochlea of the mice. And then we did a little clinical intervention study in the mice, a little intervention study where we took mice at 5 weeks and looked at hearing with ABR recording. This is ABR recording, a standard way of measuring hearing in humans, where you look at the brainstem response and the waves that are caused by the hearing. And we looked at 5 weeks, and then we gave them just a short-term treatment with NAD supplementation, nicotinamide riboside, and looked again at 6.5 weeks, 10 days later. And at 5 weeks of age, there's a significant deficiency in hearing at a 32 kilohertz, which is a common frequency for hearing deficiencies. But when we then looked 10 days later, we saw no increased deficiency in CSB from that point on. So we completely stemmed any further hearing deficit in CSB with just 10 days of NR supplementation. I've become very interested in hearing as a measure of things that relate to maybe early neurodegeneration. We know from our other mouse models that some of those that depict human diseases with neurodegeneration don't have any other features but just hearing loss. So hearing loss may be an early sign, important sign in mouse models of neurodegeneration. This is a summary, it's published, but I just want to point out that one of the interesting things we saw was that there's a deficiency in the synaptic function here measured by various approaches. And because I lost a little time, I'll just quickly say that we think that there is a specific deficiency related to synaptic function transmitted by ribeye proteins, which are NAD pocket proteins that sit there on the transmission from the inner hair cell to the afferent neurons. And we think that this is where NAD, maybe because it's an NAD pocket protein, has a specific effect and can really improve hearing. We also have such ribeye proteins in the eye, where other studies have shown that NAD supplementation is effective. So I'm saying this mainly because I want to return to the issue of synaptic transmission. And now I'll just move quickly into Alzheimer's, which we have been very interested in for now quite a long time. And as we all know, Alzheimer's is a huge and complicated problem. And in no way I'm going to minimize this and say it's just due to a few things. It's very complicated, and there are many ways of looking at the cause of Alzheimer's and the progression of Alzheimer's. And clearly, it's a very, very important problem because we don't have any treatment for Alzheimer's disease. Amongst the things that people agree on is DNA damage aspects. Mitochondrial dysfunction has become very important in Alzheimer's. And I know from many meetings I've gone to that there seems to be a consensus that not only is mitochondrial dysfunction important in Alzheimer's, but may also be a very early feature in Alzheimer's. And we have a number of results and many other publications that would support that notion that the mitochondrial dysfunction is an early event in Alzheimer's. And then senescence is something a lot of groups are interested in now as also being a main feature of Alzheimer's and other diseases as well. So when we think about DNA damage and repair in Alzheimer's, there's a lot of literature, a lot of studies, and there's, for example, pretty solid work suggesting that you have more DNA damage in AD than in mild cognitive impairment, which is a pre-stage to Alzheimer's, than in control patients. And interestingly, both for Parkinson and Alzheimer's, there is a very clear connection to DNA damage. in these diseases. Increased oxidative stress and a lot of dysregulation of DNA repair has been reported at one level or the others, but it's not clear exactly where that dysregulation lies or what the problem is with DNA repair. Interestingly, glutamate signaling at normal physiological levels in work that we did with Mark Mattson shows that ROS is introduced, mitochondrial ROS, causing DNA damage and then activating DNA repair. So glutamate signaling is the transmitter that signals in areas affected by Alzheimer's, and even normal glutamate signaling involves DNA repair. And so we can begin to speculate about what abnormal higher levels of glutamate signaling will do, causing real DNA damage problems. So we were interested in this connection to Alzheimer's and DNA repair using mainly this pathway called base excision repair, which I alluded to in one of my earlier slides. This is the way that oxidative damage, 8-oxo-G, other lesions in DNA is repaired. There's a glycosylase step, there's an AP endo incision in DNA, then there's a gap filling process with DNA polymerase beta being the main DNA polymerase involved in this process, and then a ligation process. And incidentally, this repairs a number of oxidative lesions, also less commonly studied lesions like FAPI and FAPI lesions, which are actually very increased dramatically with aging, for example. So we looked at what happens in postmortem brain, very early postmortem brain from patients. And we looked at various of these processes that I just mentioned and found that when you assay for DNA polymerase beta using this kind of substrate, which is a gapped DNA substrate that needs polymerase beta to form a cohesion between these two, then it was very deficient in Alzheimer's patients. And also the lack of this process, which is called gap filling activity, correlates very well with increased Alzheimer's from MCI controls to MCI to Alzheimer's and with Braak stages of Alzheimer's. So we thought that this was quite significant deficiency in polymerase beta. And that was now many years ago. And so we proposed based on that the step of base excision repair that's deficient is the gap filling step here, dependent on Pol beta. And in mitochondrial extracts, we were able to purify mitochondrial extracts from these postmortem tissues. And here we also, as I mentioned, base excision repair is somewhat similar in the nucleus and the mitochondria. We also looked at gap filling in mitochondrial fractions and found that this was deficient. So both in nuclear and mitochondrial DNA repair, it seems to be deficiency in AD related to this gap filling stage. Then we made a mouse, which is a 3-XTG major Alzheimer's model, deficient in Pol beta. It can't be a null for Pol beta because then they won't survive, but it's a heterozygote. And the heterozygote deficiency in this AD mouse really worsened the 3-XTG phenotype. So we got all these additional features that were not seen in 3-XTG alone, but were seen when we depleted it of DNA repair. And this includes neuronal death, metabolic alterations, and also a dysfunction of mitochondrial pathways, which we have since studied in much more detail, clearly in the complexes, mitochondrial complexes, and other functions of dysfunction associated with the lack of DNA repair. So this suggests that if you don't have proficient DNA repair, you get more or worse or faster Alzheimer's. And then we intervened with our NAD supplementation and saw very major improvement of the Alzheimer's conditions. I'll show you some of these results in some recent and more recent papers than that one. We've also looked very detailed at mitochondrial functions as a consequence of losing DNA repair. And in that work, as I mentioned, we saw defects in the complexes, defects in mitochondrial functions at various levels when you don't have DNA repair. And then also an interesting difference between male and females in this work, where we concluded that in females, there's more a metabolic shift than in males. So they go more over to fatty metabolism in females. And interestingly, of course, we would like to know why there's such a big frequency difference in Alzheimer's between males and females. And maybe it's important to understand some of these mechanistic underlying things to get at that question. So here we did this first study with NAD intervention, and we have the AD mouse model, which is 3-XTG, the 3-XTG deficient in Pol beta, and then just Pol beta alone. And what we see here is that when you look at NAD in the brain, it is lower in the AD model than in the wild type, and lower again a little bit in the AD Pol beta. But when these mice are treated with NR, the levels increase in the brain. So NR supplementation and NAD supplementation affects NAD levels in the brain. And this is quite important because I think we were the first to really demonstrate that you can have an effect in the brain by NAD supplementation. And now there's a recent study in patients with Parkinson where they also can see that when you give NR orally, this was given in water here, then you do see an increase in the brain. So we know there's an effect in the brain of giving NAD, which has been a question. And this is a measure of synaptic transmission called long-term potentiation, which is a very old traditional assay for measuring nervous conduction between different neurons in the hippocampus of the brain, 10-page gyrus, and we see that it's in the wild type, the level is pretty high. In the AD Pol beta, it's very low. After giving AD Pol beta mouse NR, we see restoration to normal levels. So this and a number of other recent studies, in fact, related to the hearing that we have done that I haven't told you about more about, suggest that this stimulation with NR plays an important role in synaptic functions. So I'll also just we're running a little late here, mention that we could measure DNA damage directly in the hippocampus. And we see that here you see H2AX increases dramatically in the AD models here, AD and AD Pol beta. And after NR intervention, there's less DNA damage as measured by H2AX. And we also looked at cell lines where we had the same result, that we looked at ADOXG and AD Alzheimer's patient cell lines, but less after NR intervention. So these studies here suggest that there is a significant effect of this NAD supplementation in aspects of DNA repair and also mitochondrial dysfunction. So we focused, and again here the work and the role of Evander Fang was very important in identifying that a major dysfunction in these neurodegenerations is the lack of mitophagy, and so we accumulate damaged mitochondria. And this has been reported in many studies that you see damaged mitochondria in AD, but how it occurs, when it occurs, has not really been clarified, and how early in the development of AD does it occur. So mitophagy here is pretty central in the process of aging, tau pathology, A-beta pathology, and inflammation. And this work here that was fairly recently published and spearheaded by Evandro's work, was a nice study where we looked at different organisms, 80 postmortem brain, iPSC cells, 80 worms, and 80 mice, for the occurrence of mitophagy in Alzheimer's, for conserved mechanisms, and how we could intervene with this. And so I think in many studies, we have been interested in looking cross species. It's important to do that because if you have similar findings across many species, then it's more of a conserved and important mechanism. And doing it this way, we had worms where you can easily do intervention kind of studies and screen. And we had mouse models where you can also do that. And then, of course, postmortem brain where you can't do any interventions. So this kind of approach allows you to screen for compounds that can be useful in intervention here. And I'll just skip over this where we showed that there is significant mitochondrial damage in 80 brains. And just mention that we've become very interested in using nematodes as model systems. And the nematodes have been widely used in aging for aspects of lifespan studies, but also for pharyngeal pumping and swimming as motor movements. But one of the things that really has become interesting for me is that they can be used to measure memory in chemotaxis kinds of studies, and that the memory deficiency we find in these studies are very similar to what we find in mice. And so it's a very much quicker way to look at behavioral aspects in memory. And in that sense, the worm has been very useful. Also in mitochondrial morphology, mitochondrial function, can the worm be really useful? And I'm particularly fascinated with that you can easily look at mitochondrial networks with TMRE staining and other approaches in the worm. And so again, the worm can be used to screen for these processes and then get much faster results. So maybe I'll just skip. We had this work was done in collaboration with Tavanorakis lab in Greece, who have very sophisticated ways of measuring mitophagy and different approaches. And we can measure it with different markers, as was done here. And then we used this approach to screen for a number of compounds. And we found that in addition to NR and NMN that we already had identified with stimulators of mitophagy in mouse models, we found this urolithin A to be a very significant stimulator. And urolithin A is a metabolite seen in pomegranates. And there's a lot of interest currently in urolithin A, and a few human studies involve urolithin A as a way to stimulate mitochondrial function, mainly in muscular diseases at this point. So this work led to identification of UA and also in Alzheimer's, and also we could use various mutants to look at pathways. And in the nematode, here is a screening, a memory screening for the compounds, identifying that NMN, UA, urolithin A, and another compound here were very important stimulators of mitophagy. And then looking at nematode mutants to identify which pathways were involved in the mitophagy stimulation affected by these different compounds. So DCT1 is involved in NMN, but not UA stimulation. And that just shows you can also get mechanistic insight in addition to screening compounds using nematode approaches. So we have done a lot of work recently with urolithin A now also, and we're doing comparative studies. So we have two strong compounds that stimulate mitophagy. they clearly work in different pathway settings. We think that UA directly stimulates the association of mitochondria with the lysosome and the lysosomal pathways in a very strong and direct way, actually. And NR a little less so, but we're using a lot of approaches now to clearly identify these functions. And urolithin A also has very beneficial effects on Alzheimer's and in the mouse models that we're studying, but clearly activates different pathways than NR does. And we find that also cathepsins are activated by urolithin A. And this is going to be a lot of work to really decipher these different pathways. But what we would like to do is to use urolithin A as well in intervention studies going forward. And then the problem with urolithin A is that the safety is not as clear, meaning there hasn't been as many studies in humans showing that there's no side effects. Whereas for nucleotide ribocyte, there are a lot of studies now showing that there are no side effects. So it's very safe to use. So this is going to be a long-term approach here at different levels. So I think maybe I should finish here. Let me just say that we also, in more recent work by Eugene Ho, who's now in Shanghai, and have studied the 2xTG80 mouse. And interestingly, the neuroinflammation is very strong in this mouse, looking at different pathways. And that is, and so here we did another intervention study and found that NR intervention here, and now we're back to using NR, really corrects pathways and individual interleukins in a very positive way. And that is work that's also been shown in other systems like cardiovascular systems, that NR, NAD supplementation with NR really has a strong effect on inflammatory pathways. And here we show it in the brain in some very specific ways. And I think, and also it actually has an effect of senescence. So we could show that when you just see senescence development in the AD model, here in the hippocampus, when you treat with NR, these mice with NR, you see a lot less senescence. That's really interesting. So NR is a senolytic at some level and maybe could be used, and again, is much safer than senolytics that are used currently are in terms of human intervention. And finally, we also have been very interested in the gas-sting signaling pathways. And this is a kind of a long story, but I think many of you might be interested in this. And what really I find very compelling is that this cytosolic DNA that signals the C-gas-sting activation pathways in many cases seem to be mitochondrial DNA. And so we showed in this work that in Alzheimer's, you get a lot of release of mitochondrial DNA to the cytoplasm, and that this is then countered by NR intervention. So right now, many groups are interested in release of cytosolic mitochondrial DNA. It's been found in Parkinson, now here in Alzheimer's. And this is not clear what the mechanism is of this release, but it is signaling to the gas-sting pathway, maybe a very early event in how mitochondrial dysfunction releases this cytoplasmic mitochondrial DNA and then activates gas-sting. Is a very important neuroinflammation, very important development of Alzheimer's. And here we did CPR here at PCR, and we saw that it's all regions of the mitochondrial DNA. So it's not just one component of mitochondrial DNA, it's various regions of mitochondrial DNA that actually are released to the cytoplasm, and a major countering effect of NAD. So I think that the DNA damage plays a key role at some level in Alzheimer's development. Maybe it plays a direct role, so you get the DNA damage at an early stage in the process, or in addition, plays an indirect role because of all of these other processes that also depend upon DNA damage, as I've just been discussing. So DNA damage is the key target, we think, and trying to further understand that is important. And with that, I'll finish now and mention some of the people here that did the work. And Deborah Croteau has been a key partner in my lab for many years, plays a major role in everything. Evandro has played a major role in this development. And Eugene Hao, who's now in Shanghai also, Sophia Lautrup, Peter Sikora developed the mouse model I talked about, and Mustafa Okur did the hearing, and I only talked a little about that work. And Jae-Hoon Park is studying AD hearing now. And many of the other people here contributed very significantly. We have enjoyed working with these groups here in Greece, the Warner clinical study. These are newer people in the lab that have contributed, and then also our colleagues in Denmark here. And the nicotinamide riboside is from Chromadex, but I don't get paid from Chromadex, I just use their drug. And a picture of the group a few years ago. We're not as many people now as we were then, but again, Deb here and Eugene is here, who is in Shanghai, and Beiming Yang is now also in Shanghai, but he works with a company there. And Evandro here, and many other people that I could talk more about. Nasir with a hearing is here. He's now in UC Irvine in California. Thank you very much. I think I'm within my time, but
Thanks a lot, Dr. Boss. So there are a couple of questions in the audience, but I have this privilege to ask the first one. Yeah. I think this from a nuclear genome kind of repair or damage to the mitochondrial dysfunction. That's really clear direction. But how about the other way around? So like mitochondrial dysfunction due to whatever reason, and then go back to the nuclear genome instability, right?
Yeah.
How many mechanisms can be kind of collected? So there are some out there.
No, I think that's a good point. I mean, obviously it goes back and forth. We clearly know that there's this retrograde response from mitochondria to the nucleus as well as from the nucleus to the mitochondria. So it's very important, clearly. Now, I mean, there's so many aspects of that, but there used to be the idea in the DNA damage field, and it goes back to work many years ago, that when you damage a cell with some exposure like X-ray or maybe oxidative damage, more importantly, actually oxidative damage, you get tenfold more damage in the mitochondrial DNA than the nuclear DNA. But that is not the case. So in all of the studies, when you have damage occurring because of this or that reason, it probably is the same level in mitochondrial and nuclear DNA. But then, so that's from the damage perspective. But in terms of this retrograde signaling, for sure, there are many ways that mitochondrial dysfunction can signal to the nucleus. Absolutely.
Yeah, because like this NAD, mitochondrial dysfunction can lead to NAD plus decrease, right? Like due to this redox balance, that can directly lead to POP1 function in the nuclear genome. I guess this is, again, there must be a lot of other mechanisms. So,
a lot of other mechanisms. Yeah.
So we should talk to you more about like in the future. But so I have a couple of questions in the audience. The first one is why defective DNA repair leads to increased mitochondrial membrane potential. I think there was a slide that I quickly flashed.
So it doesn't necessarily lead to increased mitochondrial membrane potential. Some cases it can be lower, but it leads to altered membrane potential.
I see.
I think because of all the redox signaling and stuff. So we've seen in all these cases that as a consequence of these mitochondrial dysfunctions, we also see an altered mitochondrial membrane potential.
Okay, so sometimes could be a compensating kind of mechanism, like it's just abnormal. So a lot of. So is there any new progress in the clinical trial of NR for neurodegenerative disease or NMN in that sense? So.
Yeah, there are a number of ongoing studies right now. As I mentioned, some interesting work in Parkinson's disease right now, and also some emerging work in small groups in Alzheimer's disease, but not yet big results in Alzheimer's disease. But there are a number of studies where people are trying to look at. There's also ALS, a lateral scleral. That one has also been shown to benefit from NR in mouse models in a small human study. But we don't really have large studies yet.
I see.
Major neurodegeneration.
So like related to this particular one, so that there's some debate about between NR and NMN. Actually, it turned out I know both David Sinclair and Chris Brenner. So what is the kind of your impression for whatever reason, right? What's the impression like in terms of their efficacy,
So I go to meetings with them, and we have these endless discussions.
Sure.
And I mean, this could be discussed for hours and hours. And Shinimai will argue that it's NMN is much more direct, and others will argue other things. But in our hands, we see pretty much very similar effects on mitochondrial dysfunction, DNA repair, and other things, whether we use NMN or not.
I see.
So I am not, there are many labs now who do very elegant work, the labels and other things, and really try to understand at what level the functions are of NMN and NR. But from the perspective of what we're trying to do with DNA repair and mitochondrial dysfunction, we really don't see any difference.
I see. Like now you said.
Of course, there's also the problem in the field is that many investigators have used nicotinamide riboside for a long time now, and there are many derivatives of NMN that are out there. So it's little less clear what people are using when they use NMN in their different companies. And so it's just a number of uncertainties. There will also be some new nicotinamide riboside derivatives that may be more functional. So it's a very, very big and very fast-growing field, and you will find a lot of different viewpoints on this.
I see. Okay, but at least like it's a good discussion because it's like relevant really to my country.
It's highly relevant, and because it's so relevant and because it's so hot, there's just a lot of competition, a lot going on, a lot of dispute, a lot of discussion.
Exactly. So
which there should be.
Yeah, some of them are good reasons, but some of them might be a little bit biased, but that's fine. So, so how can we rationally evaluate the degree of DNA damage and relate that to mitochondrial dysfunction in the level of organs? Another related question is about senescence. In the brain, neurons are, as we know, are not divisible. How can we distinguish the senescent one from the normal neurons?
Yeah, that's a good question, and I think that's something that we're clearly working on. I can't. I think that's a very good question, and we are looking both at neurons and astrocytes, and I think it's turning out that astrocytic, senescence may be much more important than we thought it was. So it's certainly not just the neurons. What is this one? How can we evaluate the degree of DNA damage and relate to mitochondria in the level of organs? So, yeah, that's an interesting question. So, how can we do that? We really, so with some kinds of DNA damage, like UV radiation, X-ray, you get similar damage levels in different organs and different tissues pretty much. With other things that you do DNA damage with, you can get very high individual differences in different levels. right now we have a very, I have been very conservative on that viewpoint over many years in my career and saying, you know, we really don't have big DNA damage differences in different organs and in different regions. But I've learned now that maybe that's the case. For example, we have some interesting new work that would suggest that this inner ear region is a sink for DNA damage. And that DNA damage might accumulate there and cause some of the problems with hearing that we're looking at. So there is a potential for very different damage in different regions depending on how that damage is done. And that is really not, you know, clearly understood.
Okay, yeah, thanks. There's a person in the audience raise a hand. I'd like Hong Xu, please.
Hello there.
Hi, very nice talk. So I have maybe two questions about the pyrylation of the mitochondrial DNA. What's the effect? I mean, after pyrylation of the mitochondrial DNA, what happens next? It's a repair, it's a cleavage, or what happened to the DNA?
Well, interestingly, when you have damage, it's more depleted in the mitochondrial DNA than the nuclear DNA. So maybe that is one of the things that happen. You know, there's a real interesting distribution difference between the pool of NAD in the mitochondria and the nucleus, and how that moves back and forth would very well depend on the pyrylation level. We also think that the pyrylation could post-translationally modify other proteins involved in metabolic functions of the mitochondria. I mentioned TFAM, but that's a little speculative right now. So, yeah. Okay. I don't want to take too much time because I think Evandro needs to have a chance, and we already running a little late. So maybe I'll just thank you very much for all your questions. And actually, you know, please, you can email me or something if you wanted to hear a little more, but Evandro needs to have a chance here and it's already late. So
sure. Like,
thank you.
Yeah, thanks a lot. Again, we have Dr. William Ball. So really appreciate your talk, and then let's keep contact. So our next speaker is Evandro the Fangfei. Evandro the Fangfei. You know, so I met Evandro when in the conference 2016. You know, so I was so impressed by his talk. You know, so he graduated, got his PhD from Hong Kong, and then he did his postdoc in Dr. Ball's lab, and he published for such a young age, you know, so he published more than 100 papers in Cell and Nature. It's very impressive. I mean, like, you guys know the reason, right? Because he had a very good postdoc mentor, and he got a lot of awards. Among them, the best one, I guess, is what Dr. Ball just said. You know, so Evandro is Dr. Ball's favorite student. That's amazing among 200 mentees. You know, so without further ado, Evandro. Yeah.
Thank you so much. Can you hear from me? Okay.
Yes, we can hear you. Yeah, and see you.
Thank you so much, Professor Gu, for the introduction and this opportunity. I'm really honored to be with my former postdoc mentor, Professor William Ball, to give these lectures to the society. After I finished my postdoc with Dr. Ball, I started working at the University of Oslo. I have established 3 major topics in my group. The first one is NAD in healthy longevity. We are interested in why NAD is reduced in normal aging and pathological aging, as well as why NAD supplementation was able to alleviate age-related disorders, including Alzheimer's disease. The second topic of my group is Alzheimer's disease. We are interested in impairment of mitochondrial elimination via process mitophagy as a potential risk factor of the disease. And we are also actively involved in drug development, for example, by using artificial intelligence as an approach. And the number 3, which is a major topic of today's presentation, It will take around 20 to 25 minutes. And today I want to share with you a major question we have been interested in: why aging is a major risk factor of Alzheimer's disease and other neurodegenerative disorders. So one of the hallmarks of aging, it is impaired autophagy. And we are interested in whether compromised autophagy could be contributed as an explanation as aging is the primary driver of Alzheimer's disease and potentially other neurodegenerative disorders. So with this in mind, as well as the introduction by Dr. Ball regarding NAD and mitochondria, so here I want to give you a summary on a hypothesis we have proposed that the NAD sufficient number in the cells is able to regulate autophagy, mitophagy, mitochondrial UPR, proteasome degradation, as well as mitochondrial biogenesis to maintain the balance of the healthy mitochondrial pool. However, during the aging process, reduced NAD inhibits its capacity to regulate mitochondrial homeostasis and finally may contribute to aging, Alzheimer's disease, and other disorders. And clinical as well as pathological evidence have been shown that indeed, for example, in AD, PD, ALS, and Huntington disease, there is impairment of mitochondria in through the pathological investigation. Speaking of Alzheimer's disease, reduced NAD, we argue that when this central dogma is impaired, and which causes reduced NAD, and in the condition of tau A beta and some genetic risk factors such as ApoE4 may enable this adult individual to get Alzheimer's disease. And the similar hypothesis goes to Parkinson's disease, Huntington disease, as well as ALS. My group is very interested in the development of small molecules as an approach to achieve neuroprotection and healthy longevity. And with one of the targets that is to improve mitophagy, an approach to reduce damaged mitochondria. And definitely when you're talking about small molecules, there are out-of-target effect. So we are aiming to investigate or select compounds that are majorly specific to mitophagy and with minor out-of-target effects. And today we want to share our recent study on the use of artificial intelligence to prepare drug development, especially on the development of mitophagy inducers in collaboration with University of McCall, Imperial College London, and Mount Rank AI, led by two students, Dr. Chenlong Xie and PhD students, Alice Rich-Ai. As you may already know, AI has been used in many fields, including medical fields, such as the application for biomarker development, biological target prediction, molecular function prediction. And it has also been used like precision medicine, drug screening, physical and mental health maintenance, and longevity promotion, focusing on drug development. In this recent study, I want to give you a schematic representation of the working model. So first, we have the mitophagy inducer nominates clinical or wet lab evidenced mitophagy inducers with translational potential. And then we get this algorithm being established and followed by the identification of small molecules in a library we are interested in. And with the AI-positive compounds, we evaluated its capacity to induce mitophagy in cells, the small roundworm C. elegans, as Professor Ball mentioned in his talk just now. And with the lead compound to further subject to the validation in a standard AD mouse model, XTG, and finally with lead compounds. So with this schematic model in mind, first we have a screen. We have list a couple of total of 18 mitotic inducers with translational potential, and we exclude those mitochondrial toxicant. For example, those mitochondrial uncouplers, FCCP, CCCP, among the others, because we want to do translational study. So we want the mitochondria to be functioning and healthy when we give the drug to the individuals. After the provision of those 18 mitotic inducers, we then used a machine learning approach, which involves vector representation of molecular structures, pharmacophore fingerprinting, and conformal fingerprinting. And further to generate this encoder algorithm, and finally representations of the decoder system. With this in mind, we then try to identify small mitotic inducers in a library from the University of Macau, whereby they have isolated more than 3,000 molecules from a traditional Chinese medicine resource. Why we are interested in this library? Because many of those plants being used to isolate the molecules have been used for like a cognitive treatment based on the traditional Chinese medicine history. And we thought this could increase our chance to get positive compounds, which can be further used for anti-AD research. And with the algorithm established, we first get this algorithm to get access to our virtual screen library from the University of Macau, which contains 3,200 compounds. And then by molecular representation, pretend models. And then with this approach, we are able to identify top compounds we are interested in. So here is a quantitative summary of the screening capacity. With this molecular library, which contains over 3,000 compounds, if we give a cutoff value of 0.75 with 1 as the highest match, we were able to pick up top 18 AI selected compounds. And while 7 plus 1, because 2 were very similar, were able to induce mitophagy in a HeLa-based mitochema cell lines. Mitochema is very well used, common used mitophagy reporters. And our cutoff dose was 10 micromolar. And our top 7 compounds were further subjected to the quantification of mitophagy in the neurons of the small roundworm, which is a small animal. And we have two criteria in mind. The first one is to induce neuronal mitophagy in neurons. And secondly, we're able to improve the memory in Alzheimer's disease CLX models. And we're very lucky that 2 compounds were able to fulfill those 2 criteria. We then subjected those 2 compounds to the 3XTG AD mice, which bearing both A beta and tau pathology. And when we gave the treatment of drug at 100 milligram at I think 12 months of age for 2 consecutive months, we are able to see the improvement of memory. And for the limit of time, I want to pick up a couple of data to share with you. For example, the most interesting part is the memory. I don't know how many people are familiar with memory experiments in mice. What we normally do is to subject the mouse into the water tank with milky water. And normally mouse, they do not like water. They swim and try to find a platform. This is a hidden platform. The first time it may take them a few minutes to find the platform or even longer. If the mouse is able to remember the platform after 6 to 8 times of training. They were able to find the target right away. So the short time to find the target, which indicates improved spatial memory. With this in mind, and we used this AD mice and subject to the Morse water maze. So you can see this mouse was trained for 8 times. It was still unable to locate the platform, just explored randomly. The same litimate and this mouse, it was more likely to locate in this region compared with the vehicle one, just to explore very randomly. And you can see at here, it was almost just passed it without able to locate on the platform, while our treated drug treatment group were able to find out the platform. Definitely, we had the two-way like blind approach for the experiments, and with both a female and male mice inside, you can see the quantitative results, whereby the red ones are the AD mice in the vehicle group, so they had low capacity to remember. However, for the blue and green colored, they were the same literates of mice, but treated with our lead compounds K and R. They were able to shorter the period in finding this platform close to the value of that Y-type, indicating improved performance. So what are the molecular mechanisms for those mitophagy inducers? We tracked the A-beta pathology. So both of K and R were able to reduce A-beta plaques, soluble and insoluble A-beta peptide 1 to 42. How about tau pathology? We tracked the different p-tau sites like 181, which is a clinical biomarker, 202, 205, as well as the very recent identified 217, which has been suggested as a specific and sensitive marker for Alzheimer's disease. And you can see all those markers were higher in Alzheimer's disease vehicle models. And with our lead compounds K and R treatment, all those parameters were lower compared with that of the vehicle group, indicating reduced p-tau. And we also saw a reduced tau tangles. As I mentioned to you, when you are using small molecules, we always have the auto-target effects, indicating, for example, even our lead compounds were able to induce mitophagy and were able to improve memory. However, improved memory may not necessarily be contributed by the mitophagy capacity. So to rule out the PTN, NDPV2, and ULK1 pathway based on the microarray and Western blotting data we had, this pink-1 parking pathway can eliminate mitochondrial membrane potential erupted mitochondria and then be recognized by ULK1 for elimination. So what we did, we used these HEC293 T cells, which harboring tau P301L mutation, and they have higher tau like phosphorylation capacity, for example, tau 181. And if we have a quantitative value of 1, then you can see in our lead compounds K and R treatment, we saw 50% reduction of p-tau size. However, when we giving the IAI-based knockdown of the gene pink-1, and we saw the dampened capacity to reduce p-tau size. To verify this is indeed the case, and we choose another p-tau size like 117, and we saw the same like pink-1 dependent reduction of p-tau size. So all those data indicating that the molecule induces a reduction of p-tau size through the mitophagy pathway. Here I wanted to give you so what we are doing now, it is we are using AI to further modify the structures of our lead compounds K and R to enable it to have even better biological functions if it is possible. And we are also using deep learning capacity to identify new mitophagy inducers. And a deep learning approach like this general had been shown can very quickly identify new compounds of interest. So here I want to give you a summary of today's talk. Some I didn't show, but I think they related to what Professor Ball mentioned and what my talk highlighted. We have highlighted the importance of the NAD mitophagy axis, which has an important role to play in healthy aging. While for pathological aging and general aging, this pathway may to some extent impaired or dramatically impaired. Combined with people with different disease mutations can contribute to different neurodegenerative disorders. And we also showed defective mitophagy is a risk factor of Alzheimer's disease, while mitophagy, which determine garbage clearance capacity, is a drug-able target. We have been using artificial intelligence and validated it as a fast, efficient, and useful way to screen drug candidates for Alzheimer's disease. And future deep learning techniques should be used in developing new drug candidates against aging and Alzheimer's disease. I think in January this year, there was a special issue on in Nature on Aging, which highlights the importance of different angles for in the aging field, including like autophagy as well as the NAD field. Finally, I want to highlight this Norwegian Center on Healthy Aging network we have been working on. And we are very pleased to have consistent support from Professor Wilhelm Ball as well as the others to give me advice on the development of biology of aging, this topic in Norway. And finally, I want to thank my current team members, including Dr. Chen Longxie, who was the first author of the Nature Biomedical Engineering paper, and now an associate professor in Wenzhou Medical University, and Dr. Yaya Arman, who is now an editor in Nature Aging in his London office, as well as my collaborators and my major funding, my conflict of interest. So thank you so much for this opportunity, Professor Gu.
Great, Evangel, that really exciting stuff. I hope the last time I learned a new knowledge this time as well. So for your AI, so your seed compound, right? Like you don't have a lot of seed compounds, right? So like that can reduce the, because like maybe because of this you miss a lot of real one, right? I guess.
You mean the lead compounds are?
No, not lead compound, like the seed one, the one you use to train your model, right?
I think based on the AI, since I'm not an AI person, so the AI part is in collaboration with my colleague. But I think the general way is if you provide more, your results will be more accurate. So because right now we have a limited number of mitophagy inducers, like only 18. So we were unable to use the more fancy deep learning approach.
Okay.
We can only use the basic machine learning to reduce any false positive.
I see. Like first of all, you're saying you're listing A or like spermidine or like NR or are in this list in this list. Oh, okay. Yes. That's interesting. You know, so also I see some other people in the audience. Dr. Ball, do you have any questions? To see so exciting stuff from your student, you know, so that's amazing. You're muted. You're muted.
Well, it's very exciting. I'm very pleased to see that Evandro is off on his own doing such nice work and establishing a new area here. And I wonder a little bit about these compounds, K and R, Evandro. What effects do we know what a little more about them, about what can you tell us more about them, or is it not clear what they exactly are?
Yeah, so the lead compounds and K and R, they are polyphenols. They have been shown to with antioxidative capacity, but none of them have been reported as a robust mitophagy inducers. And we are now further modify structure of K and R. So those two, they are published. K is camphophyrol. R represents refutigene. So both of them are polyphenols, and their structure are simple, so which enable us to do further AI-based modification. As you may already know, there was the dose we used for the in vitro study. It was actually very high at 10 micromolar. If we reduce to 1 micromolar, actually they were unable to show detectable mitophagy capacity in our mitochema. If you're talking to a pharmaceutical company, they are eager to have molecule at 1 micromolar if in cell culture, because 10 micromolar may be so high if you like correlate to the translation to clinical.
For sure.
So that is what we are doing now.
Yeah.
So both of them can cross blood-brain barrier. Like that's pretty lucky, so.
I think the compound R were able to. K does not.
But they also, but K also show the impact on the brain. For why?
So I guess what maybe you're saying, Andrew, is we don't. I mean, it has an effect in the brain, but we don't know exactly if it's a metabolite or something else. That's the same with what I talked about with NR. That I'm just saying. The effect is there on the brain. We increase NAD levels, but we don't know for sure if it's a modified metabolite that actually goes through the blood-brain barrier.
Interesting. Okay. So there's a question from audience also about KNR. KNR only increase mitophagy or increase all kinds of autophagy?
So at the dose 10 micromolar we used, actually we're able to check it also increased the general autophagy. Yeah, so this is not because it can also understandable since mitophagy is a subtype of general autophagy. So if you have a huge increase of mitophagy, there's no doubt autophagy is also increased. But as we mentioned, we pinpointed out it can, at least for the p-tau, this pathology, it is dramatically dependent on the mitophagy pathway. Because when we knock down the mitophagy gene PINK1, the drug-dependent reduction of p-tau capacity was annulled.
Okay. Like it's 11:30-ish for Dr. Bo, said.
Yeah.
Eventually, your morning is here, So I don't know, any question from the audience? Okay, if not, let's thank both speakers again for this great, exciting, enlightening talk, and we'll keep contact with you in the future, And
it was nice to meet you all, and enjoyable interaction. Thank you for having us.
Okay, thanks a lot. And keep contact. Eventually, maybe you should get some sleep as well.
Yes, I will. Yeah.
Yeah.
Thank you, Professor Gu, and nice to see you, Professor Ball.
Oh yeah, thanks a lot. Okay, bye-bye.
Yeah.
Yeah.
See you. Bye-bye.
Bye.